I request once again, I request everyone once again to start their cameras. With that, a very warm welcome to our distinguished guest and dear participants. On behalf of BVDU ISPOR Student Chapter 2025-26, it's my pleasure to welcome you all to today's online session titled Decoding Precision Medicine. I am Som Sapkar and I will be your anchor for today's session. The future of healthcare is rapidly shifting from generalized treatment approaches to highly individualized, data driven care. Precision medicine represents this shift where biology, technology, and analytics come together to improve patient outcomes. Understanding this paradigm, it's essential for students, professionals working in life science, healthcare research, and pharmaceutical innovation. It is now my privilege to introduce our speaker for the evening, recognizing someone who truly works at the interface of biology and computation. We are honored to have with us Mr. Sparsh Agarwal, a biotechnologist and bioinformatician from KU Leuven, Belgium. Mr. Agarwal holds a Master of Science in Bioinformatics from KU Leuven, where he graduated in cum laude, reflecting his strong academics and research excellence. He completed his Bachelor of Technology in Biotechnology with a specialization in genetic engineering from SRM Institute of Science and Technology. His research journey is deeply rooted in molecular oncology, translational research, and computational omics, with a strong emphasis on data driven discovery. Currently, he is involved in advanced research at the VIBKU Leuven Center for Brain and Disease Research, where his work focuses on genetics and multi-omics analysis in Alzheimer's disease, a field of immense relevance in today's precision healthcare landscape. Previously, Mr. Agarwal has contributed to several impactful research areas, including transcriptional regulatory networks in microglia, tumor environment interactions, cancer biology projects, exploring dormancy pathways, and cancer stem cells. He has also co-authored peer-reviewed publications in oncology and therapy development and has presented his work at national scientific conferences. We are truly grateful to Mr. Agarwal for joining us today and sharing his valuable insights. Without any further delay, I invite Mr. Sparsh Agarwal to take the virtual stage and guide us through decoding precision medicine. Over to you, sir.
Thank you, Soham. Thank you for the extremely warm welcome. I'm flattered.
Most welcome, sir.
Yeah, hello everyone. I hope everybody is good, enjoying their life. I'll not take too much time and I'll start sharing the presentation. Is it visible?
Yes, sir, visible.
Okay, so there's not much to tell about myself because Soham has already said more than I could have ever told about myself. Hello, today I'm here to talk to you about precision medicine. Basically, I'm a bioinformatician trying to sell you bioinformatics to use in your field. And I want to introduce precision medicine not as a buzzword, but as a logical evolution of pharmacology once we accept that patients are biologically different. And I think it's safe to assume everybody is biologically different here unless somebody is secretly practicing cloning. So we know that we are all biologically different. So it's only logical for medicine to also evolve towards that, towards personalized and specific medicine for everyone. I'll focus mostly on the theoretical foundations and the concepts where the variability comes from and how we measure it and what kind of data are there and how it changes drug development and how it improves drug development at the end. So before diving into technologies or diseases, we need to clarify the terminology. What is precision medicine? By definition, it is biomarker-driven approaches targeting specific molecular pathways. So it's when you use biomarkers to diagnose and treat more precisely and more in a more targeted manner. And the goal is ultimately to reach personalized medicine where you can uh, customize the treatment to each individual, but that is rarely achievable. So we usually start with stratified medicine by using measurable biomarkers to place patients into biologically meaningful groups, which can be also by ethnicity, by geographical distance, by genetic similarity, and mostly by biomarkers that are clearly stratifiable. Okay, yeah. And then we start with stratified medicine, which is a little more general than personalized medicine. And then we try to go as much as possible towards personalized medicine based on how much, what is the range of available therapeutics for that disease, and what are the different protocols available, how much flexibility you have to further specialize it for that individual. We attempt to do that on a case-by-case basis. From a pharmacology perspective, this directly affects efficacy, toxicity, and metabolism, the core outcomes that you care about. So let's start with the need. Why do we need precision medicine? Because, as I mentioned, we are all biologically different. And what are the sources of biological variability there? Precision medicine starts with that one basic observation. Genetics sets the baseline. Epigenetics modulates it, and environmental influences reshape it, and even the microbiome can alter the drug metabolism. Importantly, these sources interact. Precision medicine exists because ignoring this variability produces average drugs that work well for no one. So understanding the variability from all these factors and how they interact to create complex differences between people, between patients, is very important to enable precise therapeutic targeting. To act on biological variability, we need technologies that measure it. Several milestone technologies and approaches developed in recent years now form the backbone of precision medicine by enabling precise molecular profiling. Next generation sequencing kicked off the biotech boom as we know it by enabling high throughput genomic analysis and comprehensive mutation detection and profiling. In addition to genomic sequencing, studies with proteomics and metabolomics reveal functional states. And the relatively recent rise of spatial omics, where you are able to, it literally adds another dimension. The data went from 1D to 2D, preserving tissue context and telling us where the biology happens. And lastly, a recent approach called polygenic risk scoring. It tries to aggregate small effects across the genome for holistic predictive modeling and patient stratification. These tools don't replace pharmacology, they inform it. So I will talk about precision medicine through the context of two diseases that lead the charge for precision medicine and that I have worked on as well, which is cancer and Alzheimer's. These diseases are ripe ground for precision medicine research because they are both multifactorial, involve dysfunctional across multiple pathways across several cell types, and hence vary in causes, manifestation, and treatment response greatly by patient. Crucially, they provide contrasting examples. Cancer shows where precision medicine works best, and Alzheimer's shows where it's hardest to implement but most needed. Let's start with cancer. Cancer became the proving ground for precision medicine due to its sorry, due to its high mutational burden and well-defined driver mutations that serve as actionable therapeutic targets. The field has evolved from organ-based to molecular classification, enabling targeted therapies. However, tumor heterogeneity remains a significant challenge, driving the need for combination approaches and adaptive treatment strategies. In cancer, we don't just look for mutations, we look for drivers. These are alterations that actively sustain malignant behavior. Key molecular drivers like EGFR, BRAF, ALK, and HER2 represent validated therapeutic targets in specific cancer types. Understanding mutational signature provides insights into carcinogenic processes, while analyzing clonal architecture reveals tumor evolution dynamics over time, and the hallmarks framework helps identify fundamental cancer capabilities that can be therapeutically targeted. Once drivers exist, diagnostics become essential. Companion diagnostics, including IHC sequencing panels and fluorescence in situ hybridization, provide molecular information. Liquid biopsies can add a dynamic dimension, allowing us to track disease evolution in real time with less invasive repeated monitoring through ctDNA analysis, circulating tumor cells, and exosomes. Tumor boards provide multidisciplinary interpretation, while validated stratification markers guide the final treatment decisions for optimal patient outcomes based on the information gained by companion diagnostics and liquid biopsy approaches. So mainly targeted therapeutics encompass small molecules for intracellular targets and monoclonal antibodies for extracellular targets. Successful therapy requires a deep understanding of the affected pathways, such as if you know the pathways in detail, you could find deficiencies and problems in the pathways that you can then exploit for more targeted uh, therapy, such as PRP inhibitors and synthetic lethality in BRCA mutated tumors, exploiting the DNA repair deficiencies. Another thing very important is to understand the potential resistance mechanisms, because even if your therapy is targeted, but there are alternative pathways, bypass pathways, or secondary mutations that make it ineffective, it's pointless at the end. And most importantly, uh, and also designing combinational therapies to overcome the resistance mechanisms and to increase the precision further. Most importantly, precision medicine is not static. It requires monitoring and adaptation continuously. Yeah. I think I'm going up. Although immunotherapy is often seen as broad, it represents a precision approach requiring careful biomarker assessment. PDL1 expression, for example, remains the most established predictive marker, though with variable cutoffs. Tumor mutation burden shows promise but faces limitations, while neoantigens prediction remains challenging. Advanced immune profiling through TCR sequencing, multiplexed IHC, cytokine analysis, and gene expression signatures provides comprehensive characterization for optimal patient selection. Now we'll discuss the case of Alzheimer's disease, which provides a bit of contrast to cancer because it's pretty much the opposite of cancer. In terms of comparison, let me put it this way. In cancer, you have one, you have driver mutations. You have particular mutations that cause the cells to grow uncontrollably. But you still have a target. You have cancer cells that are growing uncontrollably and hijacking the entire system around them. So you can still try to target the cancer cells, try to kill them. Even if there is no precision medicine, what has been happening till now, chemotherapy and radiation, just try to kill something. You can still do something. But in Alzheimer's, it's the opposite. The cells are dying instead of growing uncontrollably. And it's the immune cells that cause that primarily. So in cancer, you can try to tell the immune cells to better target the cancer cells. But in Alzheimer's, we don't even know what's causing it. It's the immune system that completely fails, the clearance system that completely fails. And they themselves turn harmful, as I'll show later on. So it shows us a bit of, how a different approaches in this can be applied in comparison to cancer. So first, why is neurodegeneration different? The main problem is that the brain is hard to access. The blood-brain barrier severely limits drug delivery, and long prodromal periods in Alzheimer's mean that the pathology begins decades even before symptoms. So it's very difficult to treat it before it goes out of hand. Alzheimer's has a multifactorial etiology, as I told. We don't have driver mutations in Alzheimer's. It's multiple systems, multiple pathways across multiple cell types. Somebody's mic is on. Yeah, in Alzheimer's, you see multiple pathways across multiple cell types just going off at once. There is protein aggregation inside and outside the cells. The neurons are dying, the metabolism fails, and the immune cells go into a panic mode, creating excessive inflammation, which only makes things worse. And we still don't know which exact mutations cause these, because again, brain is difficult to study. It's very difficult to make usable model systems for the brain tissue. And often what we study in cell culturing or in mouse models don't translate to actual clinical outcomes in Alzheimer's. This is a graphical representation of showing how different systems malfunction at once in Alzheimer's. So it's much more difficult to target something because we don't know what to target. And instead of trying to kill something, we are trying to revive something, which is, as you can understand, how much more difficult it is. You can't just target anything. You have to try to rectify the problems that are going on in the microglia, the neurons dying and everything, and try to somehow clear the proteins that are aggregating. So it presents a different challenge compared to cancer. Unlike cancer, AD risk is largely polygenic. There are no driver mutations, as I mentioned, that initiate the cascade, but rather several mutations and environmental factors that accumulate into system-wide dysfunction. The genetic risk of Alzheimer's spans common variants with small effects to rare high penetration mutations. ApoE isoforms are strong risk modifiers, but not deterministic, while the rare variants in TREM2, APP, and presenilin genes are more cleanly causative, but they are rare. And GWAS studies, which is genome-wide association studies, in case you don't know, GWAS is where you study a large population of varying levels of disease, and you try to, by sheer power of statistics, by just computational power, you try to figure out which loci or which mutations are associated with higher or lower risk of that disease. So GWAS has identified more than 70 risk loci, and it's just what do you study out of this? You study all of them, but they are all rare in less than 1% of the population. And what is the common part is not really targetable as much because it has small effect. It's only used for predictive studies, for risk prediction. So because we can't biopsy the brain, biomarkers are essential. PET imaging provides direct visualization of amyloid and tau pathology, which are the two major types of pathologies we see in Alzheimer's. Amyloid is extracellular protein aggregation, and tau is intracellular protein aggregation. But it is costly and less accessible. CSF markers, which is cerebrospinal fluid, offer well-validated diagnostics, but they also require invasive collection because it's CSF. And in recent years, we are seeing the emergence of blood biomarkers as well, like p-tau 217, which offer high accuracy for detecting AD pathology and minimal invasion and minimally invasive screening potential at the same time. So at the end, an integrated framework combining multiple biomarker types enables pre-symptomatic detection, precise patient stratification, and treatment monitoring. As mentioned earlier, Alzheimer's disease is more inherently multicellular, and the genetics repeatedly point to microglia, which are the immune cells of the brain. And the current top hypothesis for Alzheimer's is that the clearance mechanism of microglia, which clears the debris from the brain, it is what fails, and that's why we see the accumulation of the proteins which are not being cleared. And we see that microglia at some point even shift into helpers to harmers themselves. They become disease-associated microglia and cause excessive inflammation, which definitely does not help. So precision medicine here requires systems biology, integrated network analysis, multi-omics data, and pathway modeling and predictive simulations rather than focusing on single directly targetable mutations and directly targetable pathways. We have to look at a more systems-wide approach, look at how everything interacts with each other, and it's very complex in brain. How do you even study everything that's happening in the brain? So that's the primary challenge in translating precision medicine to AD, and which is then why we have limited therapeutic success despite target engagement and failure of clinical trials due to difficulty in capturing brain environment in lab. We see a lot of pathological heterogeneity with mixed co-pathologies across patients because of the different kinds of pathologies existing uh, simultaneously with varied degree in the patients, amyloid aggregation, tau aggregation, and vascular damage. It makes it even more difficult. We have difficulties in target validation due to model system limitations, as nothing can easily imitate the brain system. For cancer, for example, we can, uh, it's very easy relative to other diseases because of the immortality of cancer cells. We can easily have cancer cell lines, and they help us understand a lot of the basics, and the co-culture and the organoids and everything works much better. But there's no easy way to model the brain outside the brain. The leading, like so historically, it was mostly biopsy samples or mouse models, but now we also see iPSC models, which is induced pluripotent stem cells, which are then differentiated into neurons and microglia and astrocytes, and then transplanted into mice brains to see how they actually respond to the pathology and the therapy within the mice brains. And lastly, we also have a big consideration of timing in case of Alzheimer's, where intervention may be needed decades before symptom onset, which is, you can understand how that is paradoxical. That so the it the challenges necessitate integrated approaches and combining advanced biomarkers, improved disease models, and innovative clinical trial designs. Especially the big shift in the approach is from treatment to preventative, because Alzheimer's is such a silent disease and it happens. The pathology started way before the symptoms appear. The focus is more towards prediction, towards, as I mentioned earlier, polygenic risk scores, which are just a aggregated score of how many mutations, how many risk mutations does a person have, sort of predicting how much risk they have at the end. So a lot of the research here is more about the prediction, while in cancer, most of the research is about treatment. Up to this point, we have looked at precision medicine through the diseases, and we have seen that modern biology generates enormous amounts of complex data. The obvious question now is, how do we turn all of this into something actionable? This is where computation becomes essential. This is where I come in. So computational tools, as I mentioned earlier, don't replace experiments or drugs. They just act as the glue that links genetic variation, molecular pathways, and clinical phenotypes into coherent models we can reason about. The sole purpose is to inform you better for better therapeutics. Now, no single data type explains the entire disease. So integration is how we move from association to mechanism. Just a sec. So this is where most of bioinformatics right now is what we called multi-omics integration, that we have different kinds of data from the samples or the patients or the cell lines, which is genomics, proteomics, metabolomics, epigenomics, and now even spatial dimension to all of these. We now also have spatial transcriptomics and spatial epigenomics, which can show us where in the tissue the expression is high or not, and where are the cells localizing, for example. The multi-omics data integration combines these different kinds of different kinds of omic technologies, the different kinds of data sets to provide comprehensive biological insights. Network-based approaches leverage protein-protein interactions and gene co-expression networks to to basically model the entire pathways. Like there's a term called regulon that's also being used for this. So you can, once you have all this data about the genomics and the metabolomics and the epigenomics, you can try to model how they interact with each other. You can try to see which genes are expressed more and which transcription factor is also correlating with that expression spike, and try to figure out what is the entire mechanism, the entire cascade that is actually causing, rather than focusing on one biomarker or one single protein that you're trying to target. You can try to understand the entire thing to to again better predict the resistance mechanisms that are possible, to better predict the side effects or how the cells will actually react to your drug. So
So,
network-based methods add another dimension to this. Then we have the statistical part of it. Uh, these are what we learn at the very start in the bioinformatics when you start learning, is how do you reduce the high dimensionality, how do you correct for the batch effects, and how do you correct for the missing data. So these are not your headache. These are the guy you will give your data to. But
sir,
statistical methods,
I'm so
uh,
sorry to interrupt you, but can you please explain what exactly is proteomics and genomics, so that
oh,
everyone can have, including me, like a better understanding of it?
yeah, exactly. Because the problem is I'm not very familiar with what you guys study, right? You are
Yes,
very out of my field.
yes.
So sorry, definitely do interrupt me when there is something out of your scope, because this is my part. After all, this is bioinformatics here. So genomics is, as you can pretty much kind of understand just by the name that genomics is study of the genes and how to and the mutations, the DNA sequence variations. It doesn't have to be particular mutations. It can be deletions or inversions. So genomics is studying which mutations are there. For example, you take Alzheimer's patients and you take normal people and you see, are there any mutations that are solely present in the Alzheimer's patients and not at all present in any normal person? So that's how you get a polygenic risk score. You can make a landscape of mutations. You can make a model of which mutations are predictive of risk and which mutations are in cancer. You have mutations that are driver mutations that I mentioned earlier, which are directly causative, which start the cascade of cancer. So genomics is where you study the mutations themselves. Uh, I think I forgot to add transcriptomics here, but yeah, between genomics and proteomics, there's also transcriptomics, which is the expression of the genes. There you study at the RNA level, not at the DNA level. You see which genes are expressed more, which are not. You study the promoters, you study the enhancers, and how the regulatory networks work, how the different genes interact with each other, how they interact with the transcription mechanism. And then proteomics is where you study the protein level. You see which proteins are expressed on the cell surfaces. You study antigens. You study the differences in protein makeup. You study the differences in protein folding. Like in cancer, you would have proteins that are differently folded that changes their function. So it's mostly the functional analysis about the proteins. How are the proteins different in a disease versus normal state? Metabolomics is the combination of them, how these things convert within each other, the small molecules, how the metabolism is changed. Again, in cancer, you have a large change in metabolism. The cells hijack essentially the regulation of metabolism. It enables them to grow uh, uncontrollably. It enables them to capture resources uncontrollably. Epigenomics is based on the epigenetics, where just a layer above genomics, you see how the epigenetic markers modify this, the regulatory modifications that differ from person to person, how do they play a role? And this is also a relatively more targetable part of multi-omics, because you can more easily change the epigenomics rather than actually changing the mutations through gene editing and stuff. So that also plays a big role. Is it clearer?
Yes, sir. Can I ask the audience also, all of the participants, if they are, if they've understood what was said till now? You guys, I want this to be a very interactive session. It would be
Yeah,
helpful
I think I
for
went quite
Sparsh
fast.
sir also. Yes, I hope that you guys will react also, and put any questions, or if you guys have any doubts, you can ask sir while he's presenting, if that's okay with you, sir.
Yeah, definitely. I would
So,
like to,
sir,
because I'm going too
sir,
fast anyway.
I would like to ask something.
Yeah.
Yes, sir. So, epigenomics is actually how our body maintains the proteins, how our body maintains the metabolic pathways, and how exactly our body is controlling them. So, it is basically that study about that, that how is that particular individual is controlling those things, like his metabolic pathways and his proteins synthesis,
Yeah.
etc. Okay, sir.
Yeah. So epigenetics, in case you're unfamiliar, or somebody is unfamiliar, there are epigenetic modifications, methylation and acetylation on the DNA that either turn off or turn on the genes. So the genes are there. The same genes are there in everybody. But what makes us different from each other is which ones are expressing, which ones are not. And so that's where epigenomics comes on, to see whether the genes are even accessible, are turned on or not. So like the techniques we use mostly are, the most common data type we use is ATAC-seq. It tells you which parts of the chromatin are open. Uh, I'm assuming you are familiar a bit with the euchromatin and heterochromatin, that the chromosome, the chromatin opens and closes to make and closes to make the genes accessible, and that's where epigenomics comes in. So the epigenomics makes the epigenetic tags, which is methylation and acetylation. They make the chromatin open or close. So if the gene is accessible and not just folded completely, only then will the transcriptional factors be able to access it and transcribe it, right? So this is what controls the expression of the genes, and rest I've explained. Anything else? Any question about anything I have explained before? Although I think most of it was just about the diseases themselves.
So we have another question in the chat box.
Yeah.
How do you map the polygenic risk scores? Is it an algorithm?
Actually, a polygenic risk score, it sounds like something very technical, but at the end, it's just addition of the number of genes. Like if you have a panel of 70 genes, 70 mutations that are, you make a gene panel, you make a SNP panel, and you just see in this patient how many of them are present, how many of the risk mutations do they have. And you can also have weighted addition, that okay, if there are genes, the mutations that are more predictive of risk, are more associated with risk, they will have higher weight in the PRS prediction. So the models are relatively quite simple. I have personally not worked with PRS model themselves. Like the project that I did was before PRS. I was collecting the data, and in my master's thesis, I made them into a integrated database from which you can then select the SNPs for future PRS creation. But PRS is just how many of the risk mutations do you have? That is polygenic risk score. But again, that's a very good question, because PRS is like, it's not a very robust model, as you can guess, right? PRS is a basic model. So it's only useful now because we need something. We need something to have some sort of prediction, but the goal is actually to move past PRS to actually have more precise models. I will mention AI models later on, because that's where most of bioinformatics is moving now. is that now we have so much data. We have a whole sea of all these kinds of data. We have atlases. It's literally called the Cancer Genome Atlas, TCGA. So we have atlas of data, and it's enough now that we can train AI models. We can train deep learning models to actually have more precise predictions compared to PRS, which is what we have. It's like a placeholder, but it's still effective. It's what we have in practice, especially in Alzheimer's, because the mechanisms are not known. We don't know exactly which molecules to take into account, which pathways to study. It's quite unclear, the underlying mechanisms. So these kind of models like PRS and just association studies where you don't have precision, but when you have so much data, it ends up giving you something. Like just by the power of the amount of the data and the computational models, you still get something.
Thank you, sir. And I'm sorry that we keep interrupting you, but this is a completely new topic, a new thing for us.
No, I
ये
want you to
हमारे
interrupt.
syllabus में नहीं है. Yes.
I want you to interrupt. I was thinking, please somebody interrupt. I'm going too fast. I don't have enough material.
So yes, I request you, can you please go a little slower so it be better
Yeah,
for
yeah.
everyone to, yes, understand.
Yeah, no, I knew I would need to explain more in the computational part. I went fast through the diseases because it's not really important to know exactly the particular methods in the diseases. I just wanted to point out the difference that precision medicine is not just one thing. It's more of a mindset. Is that instead of trying to just throw some drugs at the disease and hoping something works, it takes more effort, but it gives you much better results. And you'll have to understand the disease, the individual pathways, the individual cell types, how they interact with each other, what are the possible side effects, the possible resistance mechanisms, how everything would work. You will have to make networks and everything. But at the end, it enables you to much improve the clinical outcome, which I will go into later on, the actual pharmacology outputs of this. But I just wanted to show that how two completely different diseases, and both are complex, extremely complex in their own way, all need precision medicine. Like not just these two, even now I'm about to start my PhD in like the next month or so, and it would be about type 1 diabetes. That's also a complex disease. It's a autoimmune disease, and the mechanisms are still not known. But the only way to go is precision medicine. We are trying to go towards precision medicine. There is one drug that is being approved right now called teplizumab, but there's a lot of variation in how the patients respond to it. So now the next step is to try to understand what mechanisms is it affecting, what are the other mechanisms in other cells that are giving resistance to teplizumab. So in most of these diseases, the only correct way to do it is precision medicine. It's just by the time it develops, people try other things that work meanwhile. But at the end, for some of these diseases, for the complex diseases, it's only logical that you automatically go towards precision medicine because they are so complex that you have to be precise. You can't just kill all cells.
Okay, sir. Sir, I have a question, sir. Sir, you said that
Yeah.
Alzheimer's disease is actually pretty hard to research about because it takes place in the brain and brain is very complex. I mean, we cannot just study the brain itself. So what will be in the case of a brain cancer then, sir? Sir, in case of a
Yes.
brain cancer.
Very good question. We do face the same problems in brain cancer, but in the case of brain cancer, it is still cancer. It's part of one of a big class of disease, right? Of course, we know that in the case of the different types of cancers, brain cancer is one of the hardest to treat because of the same reason that it's brain. It's more difficult to target the cells there. But we still have some commonality between the brain cancer and other cancers. It's still a cancer. So it still gives us something to work with. We still know the mechanisms that actually cause cancer. We still know what is happening there. In the case of Alzheimer's, we don't even know what is happening in the first place. And by the time it and by the time we noticed, by the time the symptoms appear, it is already too late. Everything is gone haywire, all the systems are failing, the neurons are already half dead, there's so much inflammation, the vascular damage. So by the time we even know what's happening, we don't know, like, it's already too late. It's already irreversible. So in terms of Alzheimer's, it's very difficult to predict it, first of all, and it's also difficult to treat it because we don't know the mechanisms. We don't know which cells are causing what problems. So that's a very good question. But in terms of cancer, it's still cancer. We still know the basic mechanisms. Just we need to target it more. We need to specialize it more towards the brain, which is the difficult part in brain cancer. But Alzheimer's is a whole other animal in its own. It's a whole separate thing.
Okay, sir.
Because like in cancer, in brain cancer, you still know it's cancer cells that are growing uncontrollably, right? You still know what's happening. You still know you have to try to kill the cancer cells. In Alzheimer's, there is you're not trying to kill something. There is no target. It's just your own systems that are all failing at once. Right, the proteins are incorrectly being folded. The microglia, the immune cells that have the job of clearing those proteins are not doing that for some reason, and then they create inflammation, they create vascular damage. So just it's a whole cascade. It's a spiral of things happening after one another, and you don't know what actually caused it. When did it start? How did it start? It's multifactorial. It's not even purely genetic. It's environmental factors, growth factors, your growth and your genetics, your family history, all of that. So the challenge is mostly about first understanding what the disease itself is. And that's where the difference between cancer and Alzheimer's is that in Alzheimer's, we don't even know what the disease internally is. We don't know the mutations that cause it. Any more questions about the diseases?
So we have one more question in the chat box. Would ctDNA be a good starting point for precision medicine in brain cancers?
It would be, I think, yeah. I'm not very familiar with the current status of what's going on in brain cancer research, but I would assume, yeah, in terms of just like in Alzheimer's, the biomarkers we use in Alzheimer's are the ones we get from the CSF and the blood because it's difficult to go into the brain itself and get something to cross the blood-brain barrier. So yes, I think in brain cancer, ctDNA might be it might already be something that's being studied there. I'm just a bit out of touch. I've never worked on brain cancer. So if you look into how brain cancer research is going right now, I'm sure you will find ctDNA to be a part of it.
Thank you, sir.
Okay.
Do we have any other questions up till right now? Okay.
Okay. So now that we all are clear about, hopefully clear about the different types of data that are possible, which is genomics, transcriptomics, proteomics, metabolomics, epigenomics, then we have to integrate them to arrive at something useful, not just raw data. So I mentioned already the first approach is the network-based methods, where you try to make a network across them, which mutations are causing which genes to upregulate or downregulate, and then as a result, what is the protein being formed? Is there a mutation that's not causing difference in the regulation, but the protein is different? It is misfolded? Is it the protein is incomplete? So you try to understand the whole network across the different layers and how the proteins interact with each other, how the genes interact with each other. And you try to build a model of the pathway, the entire pathway, to try to understand what would be the effects, where can you block the pathway, where can you target which parts can you target, and what effect will it have. a particular kind of targeting will cause something else to go wrong or not. So that's where network-based methods come in. And like there for all of these nowadays, it's like I would say this is like the golden era of bioinformatics. As now when I am studying bioinformatics, we have dedicated tools for all of this. There are dedicated tools for network-based analysis, there are dedicated tools for statistical analysis. So if you want to try some, you want to learn or you want to see, it's not like you'll have to understand the deep statistics and go at the very basics of computation and mathematics. You can start with trying to use some of the tools and see how do they process the different kinds of data, what kind of things they do. So it's now more than ever, it is easiest to enter bioinformatics to get introduced to it. In fact, that is how I got introduced to it. I never came across bioinformatics as a topic. It was never in my curriculum. I was working in Mumbai in Tata Memorial Center, ACTREC, that's a Cancer Research Institute. And my training there for 1 year training, it was completely cell biology based. It was wet lab. I was working with cell cultures, cancer cells, and fibroblasts, and doing cell experiments, functional assays. But I also was trying to see how the analysis is performed. I was just playing around with some of the tools, and I found interest in it. And I found, okay, this is something that I like, and it makes sense. So I also came into bioinformatics just by trial and error. So I would recommend to keep an eye out at least for what is happening in the field of computational biology and bioinformatics, because right now it might feel foreign to you, but it is coming. Computation is coming for everyone, not just biology. Computation and AI is coming into every field. Every field from physics to biology to chemistry is now relying heavily on computation models to process the large amount of data that we have collected over all these years of physical experimentation. So we have network-based methods. I'll continue with statistical challenges. There are statistical uh, methods. Yeah. So the statistical models, I would not go into the specifics, but there are models that there are methods in statistics that then address these high dimensionality batch effects and missing data problems. And and more recently, we have now machine learning, where instead of going from the very scratch, you just try to get machine learning, deep learning trained on the large amount of data that we have. And it includes multimodal deep learning, feature selection algorithms, and predictive modeling as well. Yeah. So all these methods, all these approaches using different computational uh, approaches, different computational concepts, and even mathematical concepts. We have a lot of mathematical modeling as well, where we try to make simulations of the cells, try to simulate how the cell will behave if something is changed, if a protein is changed, if a protein is depleted. I will also mention it later on in one point. It's called digital twin studies. Now, a quick introduction to some functional genomics tools. So after we get the data, we make some models. How do we actually bring it to the lab? So CRISPR screening enables the high throughput assessment of the gene function through loss of function, gain of function, and base editing approaches. So you make a big model, you make a large statistical model, and we come out at the end, okay, these are 15 genes, these are 15 mutations that are highly that predict the risk of Alzheimer's, and they are expressing in microglia and everything. What you do then is you bring that to the lab. You make those mutations, you make CRISPR screens, you make them in the cells, and you see what are the functions that are being gained, what are the functions that are being lost, and how the interactions and how the behavior is changing. And we also have regulatory element analysis, as I mentioned, that this is where epigenomics is. is ATAC-seq and EQTL, where you can see which chromatin parts are accessible or not. And EQTL is expression quantitative trait loci. This is a statistical method which says which mutations are affecting the regulation of the genes, not the gene itself, not the protein itself, but the mutations that are in the promoter or enhancer that affects the expression of the genes. So this is also one approach that enables to understand what's the function of a particular mutation for a particular gene in your disease. So this reveals how genetic variants affect gene regulation. And then we have regulatory network modeling, as I mentioned,
Sir,
where you,
so sorry to interrupt you again.
yeah,
We have a question in the chat
another
box.
question. Yeah.
Please explain what CRISPR actually
Oh, sorry. Yeah. Yeah, these things are like ingrained in my brain, so I forget that. Yeah,
is. No, sir,
I forget
completely
to ask
understandable.
if you know
Yes,
or not.
yes, sir.
Okay, CRISPR, like, oh man, that's a big topic, but I'll quickly introduce. CRISPR is a gene editing tool. It is, in fact, in biotechnology, it is very much our, holy grail nowadays. Like this, it was the greatest, it's called the greatest invention in biotechnology in our generation because it enabled easy gene editing with high precision. And the scientists that who discovered CRISPR, discovered slash invented because it's so CRISPR itself, the mechanism exists in bacteria and we co-opted it, we modified it to use it as a gene editing tool, right? And the scientists who invented it or studied it, they got the Nobel Prize, I think in 2022. Yeah, 2, 3 years ago, they got the Nobel Prize in medicine for the invention of CRISPR. So CRISPR, I will not go into the details of CRISPR. It's just a way, technology to edit a very specific mutation, just create a point mutation. So once you have a list of mutations that are predictive of your disease or that are involved in your disease somehow, you can make a screen. You can just have a collection of cells where you edit all those genes, all those mutations, and you see which cells are surviving, which cells are responding to the drug, or different kinds of studies. There are loss of function, gain of function, base editing. So you can check target validation, you can check drug resistance, you can check lethality. So you can make a mutation and see if that kills the cell, or if it is a resistance related, you can see if that mutation is what giving the resistance or not. So you can have multiple applications based on the mutations. I hope that's clear.
Yes, sir.
Yeah, okay. Was there anything else in the chat? No. Oh, there's one more question. Yeah. Can AI-driven multi-omics predict drug responses like repurposing is? Yes, very much. AI is now in every part of bioinformatics and biotechnology. Like anything you have data about, you can have AI because AI, most people think, oh, AI, it's a big thing. And when we say AI, it's incorrect to say AI. What I'm mostly talking about is machine learning. It's deep learning. It's just statistical models at the end. It's just models that you train on your data and then you get a response. It's just like plotting a graph. It's just a higher version of that. And you have data, you find patterns in the data, and so that when you give it new data, it can extrapolate using the pattern that it learned from the existing data. So anything can be predicted. Yes, drug responses are one of the things where AI is being explored in, but it's a difficult field to implement because to predict drug responses, you need a lot more context, right? Because drug responses are not like, okay, you are targeting a protein, it goes and does that. The cells are very complex. There's a whole system of pathways, of mechanisms that every protein is involved in. Every gene, every uh, protein, they are involved in multiple pathways, multiple processes. So to predict a drug response, it's much more difficult because there are so many more things you need to take care of. Right? It has to be a much larger model of the whole cell, of the whole pathway. You have to understand the drug that what your drug is doing. You have to understand your drug completely. You have to understand how will it bind, which proteins can it bind, how specific it is to one protein, can it bind to multiple different proteins, right? You have to understand everything that can go right and everything that can go wrong. So to predict drug response, you need a lot more context about the target that you are using, about the drug that you are using, and everything around the target, everything involved in that entire system of pathways. You need to understand to correctly predict the drug. But yes, it is one thing, it is a thing that is being explored actively to try to predict. And as I mentioned, go past simpler models like PRS and stuff to actually have better prediction of risk and better prediction of response.
So I wanted to ask, does genetic factors?
Yeah, your voice is echoing. Sorry.
Rekha, I think you've joined by two devices. So could you please mute your other device and then unmute just one of them and then ask? Or you can put your question in the chat box. So, like you mentioned, how AI is now coming into every field and it's advancing at like a very rapid pace. Could you please share an example of how AI, or not AI, sorry, machine learning is now being utilized in enhancing precision medicine in maybe pathology analysis or treatment prediction, as Rushikesh's question was?
Well, I'm not very much in touch with the AI or machine learning side of bioinformatics and precision medicine, but as I understand, like I'm again, I'm not very much in touch of what's the current trends in this. But as I understand, right now it's not at the stage that it's already being used for predictions. It's still being explored how we can do that, first of all. Right? Like in my master's thesis, that project, there was a collaborator in in Oxford University who was doing the same thing, but from a machine learning perspective. Just like I built a database manually adding different kinds of data and manually filtering them and trying to find out something relevant by just biological understanding. But the same data was being also used for training a model to see if that can somehow predict better or just give us prediction without having to go through the entire manual curation and integration of data. So I think in almost all disease research right now, somewhere people are also attempting if AI can do, but in practice right now it's all still manual because it's a sensitive field. If you're trying to predict a drug, predict a disease incidence or a drug response, it's too sensitive to just give it to AI. It's not like ChatGPT that can just predict your language. Biological systems are so much more complex than English. So the AI that you are talking about here, the machine learning you're talking about here is much different than what we know in general daily life. It's a whole new field. It's a whole new level of learning, of machine learning, because you have to learn much more complex systems. Okay, there's one more question in the chat.
Yeah, so the addressing Shikha's question, sir. Does
Yeah.
genetics factor influence susceptibility of cancer or AD? And if it does, how can these details can be used in precision medicine?
I mean, I think that was already quite extensively mentioned in my work. So, like in cancer, especially, we see a lot of genetic effects. Uh, Okay, genetic factors influence susceptibility. Yeah, so in cancer, genetic factors are like not just susceptibility, they are causative. We know the driver mutations that cause cancer in the first place, and those are the ones we try to target mostly, such as EGFR, BRAF, and HER2 mutations. So we have like that is one of the biggest advantages in terms of cancer, I would say, that it is very much genetic and we have actionable mutations that we can clearly try to target and try to understand their pathways and how to block the effects of them. So in cancer mutations, the genetic factors are just uh, and they are inheritable. A lot of them are inheritable, but it's also spontaneous. I mean, both those diseases, cancer and Alzheimer's, are both multifactorial. There's also a genetic factor, inheritance, but there's also spontaneous factors, spontaneous mutations, and environmental influence. But yes, genetic factors do influence the susceptibility in cancer and Alzheimer's disease, and they are being actively studied. Like most of the because even if there is no genetic factors, understanding the genetic parts in those diseases can help us understand how non-genetic factors will also affect, because the symptoms and the outcomes are similar. So understanding the genetic factors is the easiest way to start understanding the disease as a whole. So that is how most of it is done. You first start with the genetic parts of the diseases, and then you try to understand how everything else works and how it would be in the spontaneous patients who don't have a family history, who don't have a genetic susceptibility, but they still develop the disease, which is the case in both cancer and Alzheimer's. In fact, in Alzheimer's, I think it's uh, 90%. Yeah, I think it's 90% of Alzheimer's are spontaneous. They don't have genetic susceptibility. They are not congenital. So, but still, there's most of the bioinformatics research, most of the precision medicine research starts from the genetic side, because we need an entry point to understand the disease in the first place, and genetic factors do give us a good starting point there.
Yeah, thank you, sir.
Where was I now? Yeah, I was in the computational approaches. Yeah. Okay, so as I mentioned that there are functional genomics approaches such as creating the mutation and seeing what happens to the cells, trying to understand how the regulation of the genes is affected, and we can network the models, model the networks, and at the end, it all leads to different applications in precision medicine, such as biomarker discovery and risk gene assessment, target prioritization. Okay. So I'll give a quick example of a typical computational pipeline that is conducted in precision medicine bioinformatics. So you start with multi-omics data acquisition from uh, usually like the databases we have now are from thousands of patients, so that we understand, we can see the patterns and it's statistically relevant. And then the rigorous quality control is performed that ensures data integrity through read depth assessment, sample quality checks, and technical artifact removal. And then we integrate using the multi-omics integration tools. It can be manual tools or machine learning tools that integrate the data, and it employs network-based and machine learning methods for data fusion. And at the end, hopefully, we have a model that can help for prioritizing what to target, what gives the risk, which patients to stratify into which group, and further, the designing of the therapy strategies. Now here in the computational pipeline, as I mentioned earlier as well, the cancer-specific analysis focus on tumor and normal comparisons and clonal heterogeneity, how the cancer cells evolve from benign to malignant, and how do they affect the other cells, and how do they change over time over the progression of the disease, and the driver mutation versus the other mutations that just like pile on, which are called passenger mutations. While in AD, the analysis is emphasized over longitudinal data, which is over time. So with aging and over time, how many years the different pathologies developed, which pathology developed when, and trying to predict and diagnose before symptoms even appear. So trying to understand the risk is more important in AD versus this is why like the in the computational pipeline here at the integration and quality control, it completely differs from disease to disease. The overall idea is the same, the overall flow is the same, but internally the tools that are used, the types of analysis that are used after integration to reach a model are so much. Like, can you see my mouse by the way, or should I use a pointer? You can't see my mouse, right?
No, I think so.
No, sir.
You'll need to use the pointer.
I've been waving my mouse for so long. You can see the pointer now?
Yes, sir.
Yeah, so just like this part where we actually have the data and then what to do with the data differs so much from disease to disease. The kinds of analysis that are done are very different across diseases because the diseases are different. They are very, very different from each other. So, but my point here for pharmacology people is no matter how much different the diseases are, if they are in the case of complex diseases, precision medicine is the same. It's more about the mindset. It's more about the approach that we would try to use more directed, more targeted data, use data for more targeted therapies. And internally, that would be different. The approaches, the considerations, the factors would be different across diseases. But trying to understand the disease and trying to use the data about the disease, trying to use the data from the patients is the only logical way to go ahead. Who's using the pen here? Like I see marks on my screen for some reason. I don't know how that is even possible, that I see lines and dots
So
being
on to it,
created
sir.
on my
We'll turn that feature off, sir.
yeah. Okay, I'm entering your side. Please forgive me for any mistakes that I say here. I'm not very familiar with the actual drug side of this, but I'll still go through the overview of how precision medicine fits into the and how it compares to traditional stuff, traditional medicine. So we shift the perspective from biology to pharmacology, how molecular stratification changes target selection, reduces clinical trial failure, and alters how drugs are approved and priced, especially. And precision medicine succeeds only when these insights survive the translation from data to drugs to patients. So for pharma, precision medicine is pragmatic, practical. It's it's supposed to be just natural, like, okay, why not? High failure rates, it's mostly because high failure rates are unsustainable, and pharmaceutical companies embrace precision medicine primarily to reduce the clinical trial attrition rates. And some studies, I mean, this is more established that in the current approaches, the traditional non-precision approaches over the last years, we see that there's only 10% success rate of clinical trials. Over 90% trials fail. But according to some predictions and recent data, precision approaches can almost double or triple that success rate and make the clinical trials more efficient, shorter, and more targeted with more success as well. And precision approaches also improve the responder ratios. This data set that I've used here is from one of the studies. I have the source if you want later on. But like this is about cancer that the traditional chemotherapy had a 25% responder ratio overall, but one of the targeted cancer therapies is now at a 70 to 80% responder ratio. So, and this is a no-brainer that if you target more specifically, if you target very individual pathways that do not cause enough side effects, that do not just kill all your cells, people will respond better. There will be less side effects, there will be less uh, unknown effects, and there will be a more precise targeting of just what you want to target. Biomarker-driven trial designs include adaptive designs such as this also, yeah. So biomarker-driven designing also enables us to make adaptive designs. You can, uh, especially where blood biomarkers and those are possible, which are not as invasive, you can assess repeatedly and change your protocol throughout the therapy duration. It doesn't have to be fixed. So that gives a lot of advantage as you can continuously, it's iterative. So it enables adaptive designs, it enables enrichment strategies, which is pre-screening for biomarker positive patients, and platform trials as well. I'll go, I have some more examples later on the different kinds of trials. So I'll not mention it here. Yeah. And risk mitigation through patient stratification, toxicity prediction, and earlier decisions provide substantial financial and strategic advantages because again you are more precise in who you are giving what therapy to, and you can already predict how much they will respond or not respond. For example, like I have a very recent example for this. As I mentioned, I'm about to start a PhD in uh, type 1 diabetes. So the current research in that is about the one drug that is being approved is teplizumab, but there is a lot of variation in responders and non-responders. And in the recent paper from the lab that I'm joining, they have made a genetic model. They have made a gene panel, 13 genes for responders, 13 for non-responders. So they are homing in on particular genes that might uh, determine whether the patient will respond to the drug or not. And then we are trying to study, okay, which cells express those genes, which is neutrophils primarily, and we are seeing how the neutrophils and those genes in neutrophils affect or provide resistance to the drug, and how can we further modify. So if we succeed in that, for example, you would have a model of genes and expressions that can already tell you whether the patient will respond to the drug or not. And if you can see that they have a less chance of responding, then you will have other uh, therapies to combine it. You will then from the get-go know how to use combinatorial therapeutics to ensure that they respond, or try another therapy that has a higher likelihood of response. So it mitigates the risk a lot. You already know, you can predict the response, you can predict the toxicity, and uh, make the crucial decisions early before it's too late. Now, this is again a continuation that, according to a recent study, that that in precision medicine, leveraging genetic evidence, genetically validated targets showed a 2 to 3 times higher approval rates for clinical development as well compared to non-validated ones. And perturbation screening through CRISPR and RNAi, which is gene knockdown rather than gene knockout, and small molecular screenings. It enabled target discovery in disease-relevant contexts. So biomarker-based segmentation defines molecular subtypes and identifies likely responders, while traditional medicine approaches facilitate translational medicine approaches facilitate iterative learning between clinical observations and preclinical validation. So this is I'll go through some of the different types of clinical trials or how precision medicine affects the possibilities in clinical trials. So precision medicine enables us to employ innovative designs such as basket trials, where you can have one drug across multiple cancers, or you can have umbrella trials where one cancer but multiple drugs targeting different pathways or different antigens, different molecules in the same kind of cancer. And you can have enrichment strategies, which is again pre-selection of likely responders. So this enables the clinical trials to be much more efficient in terms of you have one clinical trial for many different drugs or many different cancers. So you can have, it's sort of like a combination. It's a aggregation of multiple clinical trials, which again also increases the success rate, because you are trying multiple approaches within the same clinical trial. It increases the success anyway. Yeah, these approaches. These approaches represent a significant evolution from traditional designs, moving from unselected populations to with low response rates to biomarker selected patients with high response rates, enabling more efficient and targeted clinical development. So at the end, precision medicine doesn't end at science. Bringing precision medicine to market involves complex regulatory and commercial considerations. The companion diagnostic development requires analytical and clinical validation with synchronized drug development timelines. And there's also the part of regulatory bodies. Regulatory agencies like FDA have specific expectations for biomarker validity and clinical utility. And then there's the ethical layer. There's the question about equity of access, genetic privacy, and informed consent complexity. And there are also solutions to there are challenges in terms of pricing and accessibility, which are now being explored, addressed through innovative reimbursement models such as outcome-based pricing or population or indication-based pricing or risk-sharing agreements, where you can uh, reduce the price by basing it on the outcome or the likelihood of success. So if you have a particular mutation and that tells you that the drug is much more likely to affect you positively, you would agree to pay more price for it, right? So this also makes it easier to have a dynamic pricing. There are many different approaches towards dynamic pricing to make it more accessible. But at the end, successful implementation of precision medicine requires balancing regulatory requirements, ethical consideration, and sustainable commercial models. And there are more barriers such as data accessibility and sharing. So this is one of the big problems, that you have a lot of data being generated across different institutes, but it's protected by the data protection act such as GDPR in Europe and HIPAA in US. So you need to also see what is available, what kind of data sets are public, or you have to have collaborations across institutions to get accessible data. And there's also the biological complexity, which poses one of the biggest barriers, that even though it sounds easy that, oh, you model the disease, but you know how complex the disease is, how complex the biology is. There's always something that you will not know, and then you will find out, and then you will, in the next time, you will account for that. So biological complexity is the biggest barrier in precision medicine, because of the incomplete understanding of the mechanisms and nonlinear interactions between the biological systems. And as I mentioned earlier, network analysis is one way to tackle this relatively, that you try to model the entire system rather than just the target protein or gene that you want. Also, the forever problem of clinical translation timeline. It takes 10 to 15 years to uh, get a biomarker validated and uh, trusted enough to be moved towards translational work. That is one of the biggest problems in precision medicine because it is precise, it takes time, obviously. And there's also, again, the cost and access equity, uh, especially the geographic disparities and the healthcare infrastructure differences. So addressing these challenges requires coordinated multi-stakeholder approaches across institutions, across governments, especially the requirement of government initiative is very highlighted because it's not easy to bring these to everywhere. Like the drugs that are being developed here in Europe, the cutting edge technology will take a lot of time to reach India, and even there might need to be changes to the whole system, to the whole therapy according to Indian genetics. So the government needs to take initiatives to make it more affordable to practice precision medicine, to research precision medicine that is more relevant to your population and your patients and your particular disease phenotypes, because cancer, Alzheimer, all of them vary across populations a lot because the genetics vary a lot. Our gene haplotypes are very different from Europeans and Africans. So precision medicine is also very much restricted to geographical region, to ethnicity, because it varies uh, in effect. So it has to be localized, it has to be through government initiatives. At the last, I would also like to highlight some emerging trends in precision medicine, which also address some of the limitations that we saw earlier, the barriers and the challenges. So, for example, federated learning is where multiple institutes can have a shared computational framework, a mechanism, an agreement to share data, or federated learning exactly essentially is uh, the data itself is not shared, but the model is built together. I will not go into details, and I've also not had much of an experience with it, but there is a approach that multiple institutes can train models and do the computational research without the data sharing. And this addresses the problem of data privacy. So, and also enables multi-center discovery across different populations as well to better understand the diseases, and especially for rare diseases, the collaboration is very much important because it's very distributed and very few places research for the disease. It's important to collaborate so that we can actually even have enough data in the first place to do something. Like for cancer and Alzheimer's, there are large data sets, there are huge data atlases, but for rare diseases such as type 1 diabetes, there's not enough data. So what they have done is they have made a data set with, I think, 31 institutions across Europe and US, and like they've made a one single data warehouse called Innodia that manages the data and distributes it by limited access so that they can even start with the computational part in the first place. There's also, of course, AI multimodal models, as I mentioned, that integrate the genomics, imaging, and clinical data for comprehensive prediction. And this is still emerging. It's, as I mentioned in response to the question, it's not there yet, but there's a lot of active effort in bioinformatics to try to get some computational power of AI to make better, more precise predictions. Yes, this is also, I mentioned a bit of this earlier. There's another very upcoming research aspect called digital twin development, where you try to model the genomics and the whole system for the particular patient. So you get the data from a patient, and then you try to model how their cells, you try to model that patient's cells and tissues and see how the drug or how the treatment would work on that specific patient. You try to predict that by actually simulating their cells computationally. So this is also something very exciting that's coming up, which is digital twin development. And this, of course, enables safer testing and more better, just another layer of, validation before you actually go to the patient. And there's the next gen biomarkers using the single cell analysis, single cell multi-omics. So single cell multi-omics is right now one of the hottest topics in bioinformatics. This is how most of the research is being done. And this shift was made only a few years ago, that now we have much more accessible single cell data sets where you have the gene list or the mutation list from each cell. You have the expression of genes, expression data from each cell. So you can try to subcluster the cells. You can try to see which different kinds of cells are expressing what, and whether in a particular cell type there are further subdivisions. For example, in cancer, you see normal fibroblasts, but you also see cancer-associated fibroblasts. The fibroblasts are turned evil by cancer. Like cancer recruits the fibroblasts and makes them help it instead of fighting it. So you can, using single cell technologies, single cell analysis, you are able to identify exactly what changes are being made within the cells across the different cell types. And you have spatial transcriptomics now, which is also very recent. And you have dynamic biomarkers for real-time monitoring, such as the blood-based biomarkers, and you have composite biomarker panels. So these converging technologies using computation, using different kinds of sampling, different kinds of diagnostics, different kinds of statistical approaches, different kinds of mathematical approaches, and computational modeling as well. The convergence of these different approaches promises to transform precision medicine capabilities through federated learning, AI, ML, digital twins, and biomarkers. Okay, so we are at the end of the presentation. The summary, let's just quickly summarize the whole thing. So precision medicine represents a fundamental paradigm shift based on biomarker-driven approaches, matching therapeutics to individual patient characteristics or to patient group, characteristic of a patient group. The approaches differ greatly by disease, as I demonstrated using cancer and Alzheimer's, where cancer leads the field because of the presence of actionable mutations and accessible tissue, while neurodegeneration faces unique challenges with the blood-brain barrier, the long prodromal periods, and complex multifactorial etiology, as I mentioned. And the approach, precision medicine, uh, the approach offers enhanced efficacy and reduced toxicity with success stories like EGFR and PARP inhibitors demonstrating potential. I had slides for a few of the case studies, but I omitted them because I thought it was getting too long. But yeah, there are already success stories in cancer and Alzheimer's that have been shown to improve targeting and improve potential, improve success of the therapies. However, significant barriers remain, including data accessibility, biological complexity, and equity issues. And the key takeaway is that realizing precision medicine's full potential requires continuous technological advancement, multidisciplinary collaboration, and thoughtful implementation with multi-institute and government initiatives especially. Thank you.
Thank you so much, sir. With that, we will have some questions from the audience. The participants may post their questions in the chat box or directly ask to sir by unmuting your mic.
Whoever has a question, they can raise their hands here and then we can ask you and then you guys can ask your question. Or if you're comfortable in asking the question in the chat box, you can go ahead with that also.
Hello.
Hello.
Am I audible?
Yeah,
Yes.
go ahead.
Yeah, so good evening, sir. I am Tanmay Kulkarni. I am a sixth year pharmacy student. So I'm currently doing my internship at TCS Research in Hyderabad. So currently I'm also working around the QSP modeling in prostate cancer, and my topic of research is the in silico analysis of treatment responses of solid tumors.
Okay.
So it is somewhere in center with precision medicine only. So, but I had faced this one barrier that what we study in the course of PharmD is completely different that from bioinformatics. And the QSP model which I'm doing right now in prostate cancer, so the tumor microenvironment, the regulatory T cells,
Yeah.
yeah, everything. So all of that is completely different. So I had a barrier. So I had to start from scratch. So, what barriers we might need to overcome as a family individual to start a journey in this field? So, my way of overcoming this barrier was joining TCS because TCS works in precision medicine also. So, that was my way. But what can be other ways of proceeding as a family individual in precision medicine? And what other skill sets we might need? So, a master's probably or some other skill sets. So, what do you advise on that?
Yeah, I think in the chat also there's the same question from someone else.
Yeah,
Yeah, that how do we start in bioinformatics?
bioinformatics.
So I am myself not very familiar with what you study in PharmD, first of all, so I can't really give that much relevant. But as a like I'll answer the question that how to get started in bioinformatics and how to bridge. So you don't really need coding skills or computational skills or anything to get started in first of all. As I mentioned, we are now at the time when most of the analyses are possible through just there are websites, there are portals that where you can just explore, example data sets. There are you can have you can see, you can play around with sample data sets, just short toy data sets, and see how these things show up. What are the different kind of data? There are of course a lot of online courses on Coursera and everything for introduction to bioinformatics. I took one back when I was far from bioinformatics. I was doing cell culturing in my bachelor's. I still did a online course in Coursera, and it was a very good course, Introduction to Bioinformatics. It was, I think, a 3-course specialization or something. There are a lot of ways to get introduced to bioinformatics, but you don't have to go deep. You don't have to go from scratch, start learning a coding language or anything. The entry point should be the conceptual part, that okay, what kind of things are possible in bioinformatics, what are the kind of data that we have, what are the kinds of analysis it can give, what kind of information can it give about your particular disease. Sorry. So you can explore some tools, you can explore some online courses. It's a very easy field to get into. Like on LinkedIn, there are a lot of good researchers and people who are in bioinformatics who post especially about introduction and the different kind of pipelines. I follow one person called Ming Tommy Tang. Let me add it into the chat actually. Yeah, I've added this to the chat, the name. So this person, he's a researcher in bioinformatics, and he's very, very active on LinkedIn. He also has a newsletter, and in like he regularly posts about how to start into bioinformatics, how to. He also posts about different introductory courses about in bioinformatics. Okay, there's another question. Which tools would be good as beginners? Oh, I mean, it depends on what you're working on. For the particular kind of disease that you're interested in, or a particular kind of analysis, are you interested in the disease studying or the drug response prediction? So it depends on that and what you exactly want to. Like bioinformatics is a big, very big field. There are multiple layers to bioinformatics. There's no universal tool that I can say is good as beginners. It's mostly explorative. Just try to search. Okay, if you're working on one disease, try to search what is happening in precision medicine, what kind of approaches are there in that disease, in that particular drug related to. those cells, what are currently being explored, and you will automatically come across workflows that are done that are being used. How can students like us get involved in multi-omics process projects or research? Well, depends on what involvement is. Like, of course, you can't just go and do a multi-omics research because that requires bioinformatics background. But and I'm not very familiar with uh, how from pharma you can join. But what do you mean by multi-omics projects? Like, uh, typically what happens, for example, here is what I see. There are laboratories that are studying a disease. They have people who are working with the drug. There are people who are working to understand the disease, and there are people who are doing the computational part. It's usually multi expertise collaboration. So like what you can do is try to find labs where they have a computational aspect as well. That is also how I got introduced to bioinformatics, because the lab I was in, there was a PhD student who had computational background. So I started asking him what he's working on. I started helping him, and I got introduced to the different kinds of tools that he uses, the programming languages that he uses, and I decided to come for a master's. So it's not, you don't have to go into multiomics research head on. You just have to find labs or researchers who also have a computational aspect in their lab, who also have other projects or who have bioinformaticians in their lab. So then when you join there, even if you are only starting as your field as just the drug development part, you will of course interact with them, you will collaborate with them, and you can slowly get introduced to what they are doing, how they are processing, what kind of analysis they are doing. So that is my advice to get started in bioinformatics, is to find some place that has bioinformatics and pharma together. And especially you will find mostly in the complex diseases like cancer and Alzheimer's and type 1 diabetes, the non-pathological endogenic diseases that are much more complex. There is where precision medicine thrives most, because and it's also needed the most. So in these kind of diseases, you would all often find people who are embracing precision medicine as well in their research.
Thank you, sir. Any more questions, please?
So I had a question. It's not really a question. It's just I thought your session was very meaningful. Just one thing, the digital twin development. I have never heard of that. Could you please elaborate on that a little bit? It sounds quite interesting. I just don't know what it exactly is. That's one of the emerging trends, as you mentioned.
Yeah, actually, I'm also not very familiar about digital twin. Specifically, I've not really come across any studies with it, but like I have heard about it. It's a very, very upcoming topic. So, but what basically it means is again similar. It's basically an evolution of the network modeling itself that you have now. Once you have a multimodal integration, you have you know digital twin only works when you really have already made the models about the disease, you know what to look for. But in future we will have that for a disease. Let's say you have the mutations that affect the risk, you know which genes, which genes are expressed higher and lower, you know the entire pathway that is being modified. Then you can try to model it all together for a so the network modeling is where you try to understand it in the first place. Then you apply that model to a patient. You see, okay, in this patient, I see you take their DNA, you do the sequencing. You see, okay, these are the mutations that they have. These are the genes that are being expressed in their ones. These are the risk factors that they have. You see their environmental factors, and you try to model using computation that how your drug or your intervention will affect that particular patient. It's a very upcoming approach to try to model just the next step. Once you know about the disease, once you have models, then you can try to model the entire response of the patient itself before actually giving them the drug.
Thank you, sir. Any other questions? I think we have another question in the chat box.
Yeah, in the chat. No, that's not what I meant. Yeah, the question is that is precision medicine only effective against serious diseases like cancer, Alzheimer's? No, that was my response to how do you get started in it? That like it's most prevalent because it's most needed. For common disease, for bacterial and viral diseases, you don't need as much of precision medicine because you have more, like the targets are already quite precise. It's external agents that are coming into your body and you can target them. It's much easier to target them. So you don't really, it's already precise relatively. But for complex diseases, especially the ones that are endogenous, that to your body, that are not from external agents, that's where you need precision, because if your immune cells are acting up, if it's an autoimmune disease, you can't just kill all your immune cells. That will kill you, right? You can't kill all your immune cells. You can't, in cancer, you have chemotherapy and radiation and those that just kill all fast growing cells, but it has so much side effects because you are killing a lot of your stem cells. You are killing a lot of cells that you don't want to kill other than the cancer cells. So the stakes are much higher in these diseases that are within your body, not from external agents. The stakes are much higher and there's much more need to be precise when targeting because it's your own cells. It's not a virus or a bacteria that you're targeting, it's your own cells that you have to target. And for example, in Alzheimer's, you don't have a target. You're not supposed to kill something. The cells are dying and you're trying to revive them. How do you do that without precision? There is there is no logical way to even go without precision medicine. There's no answer before precision medicine. There's nothing before precision medicine. You are trying to revive your neurons. You're trying to make your microglia, your immune cells behave correctly. How do you do that? You can't have a drug that does that. You have to understand what is going wrong in the first place. You have to understand why is it going wrong. Only then you can try to modify something that enables it to work properly. So it's not that it's only effective against serious diseases. It's that and not just serious diseases. By cancer, Alzheimer, I don't mean serious disease. I mean endogenous diseases, more complex diseases that are multifactorial, that depend on multiple factors, that depend on complex systems, and that are in our own cells. There we need precision medicine most, which is why the largest amount of precision medicine research is being done in these diseases. So if you want to get started in precision medicine, if you want to get introduced to how bioinformatics plays a role in drug development, these diseases will be the hotspots. The research happening in these diseases will often involve precision medicine by default. So those are the places you would look for introduction to this.
Thank you, sir. With that, I would like to call upon, invite to the virtual stage Pooja and Tanvi, the president and the secretary of ISPOR BVDU student chapter for year 2025-26, to present a vote of thanks.
Hello, am I audible?
Yeah.
Yeah. So first of all, I would like to thank you, Sparsh, for such an engaging and insightful session. Precision medicine is something very new for me, and I have realized one thing that I need to read more about it to understand it in a more better way. I truly appreciate your clarity and the clarity and depth that you brought to the session. And at least it has given me that valuable perspective and a better understanding that how precision medicine can change the therapeutic landscape of certain diseases. Uh, Currently, you mentioned only cancer and Alzheimer, and I think that I should read more about it to explore more about it. And then I think that in future I can connect with you to ask more questions about it. But right now I have just one question. The slide in which you mentioned about the reliable biomarkers. And you said that AI can help or predict in identifying the reliable biomarkers for precision therapy, but there are always chances of error. So how can we I don't know how to phrase it, but I mean, what role does it play in identifying the reliable biomarkers for precision therapy, and how much reliable it is to understand that, yeah, yes, the biomarker is pretty much whatever it has identified is pretty much correct or not?
Okay, so the computational part, which is the main part of precision medicine, is actually at the very beginning of the research pipeline. So that's just a way to, like, instead of throwing a dart in dark, just randomly picking targets and trying different things, you can narrow down what you're looking at. That's where it comes mostly. The prediction is for narrowing down the millions of genes that we have, the thousands of genes that we have, to try to find out, to try to narrow your search area. Then once you have some predictions, either by manual statistical analysis or by deep learning methods, then you try to first validate them in the cell lines. And then if you see differences in cell behavior based on them, I mentioned CRISPR screening and stuff, where you edit those genes, you edit those biomarkers, and you see if the cells are behaving differently after that. And then you use mice models, or even before that, there's co-culturing, and there's like you culture the different cells together, the cancer cells and the normal cells together, and see how removing the biomarker works, and how it makes a difference in the response and everything. If that gives positive results, then you go to mice, you xenotransplant the cells into the mice and see if there's a difference. And after all of that, if everything works, if there's a positive response in mice, then you go to clinical trials, then you see how it would work in humans. So it's not at the end, it's actually at the very, very beginning, but it greatly reduces the search radius of the targets that you explore in the first place. Most of biological research historically has been trial and error. You just try random molecules and see if something kills something, right?
Yeah, right.
Precision medicine is about first knowing what you are trying to target. That enables you to better decide how to go about it. It makes the whole process faster. Just with an additional step, it makes the rest more efficient.
Okay, understood. And one more question, that you mentioned that at the very beginning of the session, that precision medicine is evolving, and many of the websites or platforms, I would say, such as ISPOR, one of the trend is precision medicine, which on which we are conducting the session. So, what according to you, in the 5 to 10 years down the line, would be the major therapeutic area from where we will benefit from precision medicine?
As in which disease, or
Yeah, as in which disease only? As in, it's not only limited to cancer or Alzheimer, right?
yeah,
But
no, it's not.
yes, but what would be the major therapeutic area where it would be applied 5 to 10 years down the line?
I mean, it's not limited to these diseases, but these are where it is explored most rigorously. So I would say, and not just cancer, Alzheimer's, I just mean the complex endogenous diseases. I use cancer and Alzheimer's as an example because those are the two most studied ones. But there's also type 1 diabetes and other rare genetic diseases where precision medicine is being explored. So, I mean, I can't predict when somebody will have a breakthrough in which disease, but it will be most likely in one of these diseases rather than pathological diseases, because this is where it has most effect. This is where it's most needed in genetic diseases, in multifactorial diseases.
Okay.
I mean, I hope it is in cancer or Alzheimer's because it's very much needed there. Those are my two fields, and I know how much we need something to come out of precision medicine there, but it will be these diseases, not really pathogenic diseases, because there's not much precision medicine required. We already know how to cure a lot of the pathogenic diseases, and it's
Yeah.
side effect free. There's not much to add there, but precision medicine comes in when it gets out of hand, when you don't have just one target that you need to kill. You have so many things, and so many things can go wrong. That's where it shines.
Okay. One last question, that are there any algorithm to design drugs that target the subcellular components such as mitochondria or something subcellular component in that?
I mean, like it's
I have
there
read
for
your
all of it, right?
paper on mitocurcumin that is
Yeah.
published. So,
I mean, I'm not aware of any particular ones that target mitochondria, but I mean that's not really a thing that, oh, there is a specific model for this and that. A lot of research is going on at every level. Different kinds of targets are being explored, and different kinds of models are being explored to predict targets for them. But AI or machine learning models in precision medicine is a very, very new field. There hasn't been any real outcomes yet. Most of the research that I came across is still exploring how to start in it. Can we even start having some models? Is there some success? Like it's still very recent. AI has been a part of bioinformatics as a whole, but in terms of the drug development, it's too high risk. The stakes are too high to just trust something with less accuracy, which is why there hasn't been enough, you know, risky trials with it. Like people are very cautious about having machine learning because the biggest problem in AI in machine learning is the black box problem, which is being discussed all over the world. That when you train a model on a data set, you don't know exactly what is causing it to predict something. You don't know what is happening inside it. It's just
mhm,
it takes input and it gives you output. Even though the output is correct, if it's accurate, you still can't trust it because you never know when it will fail.
mhm.
So it's in terms of medicine, AI is a very, you know, delicate field, I would say, and it should be very delicate because it's not an easy thing to model the complex systems. And even if there will be something that correctly predicts the targets and the responses, there will be so much scrutiny of it. There will be so much validation required after it to make sure it is correct, to make sure it is accurate, and to make sure the markers are correct and they are working. But like till now, I don't think there's much real outcome in terms of AI models.
Probably I need to read a lot about it.
Me
But
too, actually.
yeah, but once again, thank you so much for sharing your expertise with us, especially and for such an enriching session. I thank you from the whole ISPOR BVDU team.
Thank you for inviting me.
Yes, thank you so much. So I keep calling you sir, sorry, Sparsh,
It's okay.
for a very meaningful session on decoding precision medicine. And as Pooja rightfully said, that this is quite new for us. We've never um, had any sort of discussion on this before. So I think it was quite daunting for all of us and the participants to understand maybe all of the aspects to it. But you did explain it very well. Thank you for that, and thank you for also staying so patient throughout the session. I know that we kept interrupting you. So thank you for that. Over to you, Soham.
Yeah. At last, summing up, thank you so much, sir, from my side also for such an insightful or enriching session. Like the explanation you gave about the precision medicine and how it is connected through computational data integration. So it has truly helped us understand the how it is shaping the future of the healthcare medicine sector. So once again, I would like to extend a heartfelt gratitude to our esteemed speaker, Mr. Sparsh Agarwal sir, for sharing his expertise, our faculty advisor for providing a chance to arrange such an insightful session, and all the participants for their enthusiastic presence today. We hope this session inspires you to explore, question, and innovate in the field of precision medicine. With this, we come to end of our today's session. Thank you so much for joining all of you. Have a great evening ahead.
Before everyone leaves from the meeting, I hope I would request everyone to turn on their cameras. We just want to take a quick photo. I hope everyone can hear me. Can we all switch on our cameras, please? Tejal, Shikha, I guess. Are you guys ready with the camera? Tejal, can you hear me?
Yes.
Okay. Yes. Are we ready, everyone?
हो गया क्या?
Done, Tejal?
Done?
Okay, thank you.
Okay, yeah. Thank you, sir.
Thank you
Done.
so much, sir.